Hello, my name is Isadora Bordini, and today I'll be talking about scale for pest management research. This is an important issue that affects the quality of our research and our recommendations as a result. So let's review the key elements of our IPM program. So you know that our IPM program is focused on the control of our two key pests, whitefly and lygus bug. We depend heavily on selective technologies such as Bt, cotton, and selective insecticides. These selective technologies help us to conserve key whitefly predators, which are minute parrot bug, Drapides fly, big eye bugs, crab spiders, green lacewing larvae, and Colops beetle. These key whitefly predators help us to biologically control whitefly, and some of them also help us in the biological control of lygus. But when you talk about selectivity, about insecticides and traits being selective to our key natural enemies and many other arthropods, how do you know that these technologies are actually selective? Well, as many of you know, we do field trials. We do these evaluations to determine selectivity of insecticides and traits. But a very common question asked by you guys when you visit our field trials is about plot size. You want to know if plot size is large enough to account for insect movement between plots that can mask insecticide or trait effects. You had the same question about plot size. So you wanted to determine what was the optimal plot size to measure insecticide and trait effects in non-target organisms in Arizona cotton. So we established 3 plot sizes: small, 12 rows by 40 feet, medium, which was about double the size of the smallest size, and a large plot size, 4 times larger than the smallest plot size. We established controls with well-known impacts on the arthropod community. The goal of these controls was to determine at which plot size or sizes you would be able to detect these known effects and to validate our experimental design. We had water as our negative control because it does not impact arthropods. We had a positive control acephate, which is a broad spectrum insecticide that kills the majority of arthropods in our system. And we had a selective insecticide, Sivanto, which, because it's fully selective, it won't impact arthropod community. We did this experiment for 2 years. We sprayed 3 times at the highest rate, and you collected more than 100,000 arthropods. You sampled our key pests, and you collected more than 27 different groups of arthropods through sweep nets. Next, let's look at some data. Here I have insect days for Geochris or big eye bug, and you'll be only focusing here on the main effect of insecticide over all plot sizes. So here's Sivanto and the untreated check, and as expected, because Sivanto is fully selective, there was no differences between Sivanto and the check. And you have acephate here, which dramatically reduced the abundance of big eye bug because it's a broad spectrum insecticide. And this chart shows that our controls functioned. And I'm showing you here only one example, but this was consistent among all taxa. Next, let's continue looking at big eye bug abundance, but let's focus only on the main effect of plot size. So here we have the small, medium, large, and there was no effect of plot size. Here's another example with minute parrot bug, and again, no effect of plot size. And there was no ordinal pattern such as small, medium, large as one would expect. Here's another example of Drapides, and again, no effect of plot size. So you reviewed individual abundance, you check some example, but how about the whole arthropod community? We know that the arthropod community is composed by our key Wi-Fi predators, but there are many other species. So you wanted to know what happened to this community regarding plot size. And to answer this question, we did principal response curves. Many of you are used to seeing our principal response curve charts, but I'll be giving you a brief explanation. So basically, principal response curves describe temporal changes in community composition between a treatment and a control. These changes are the community response to the free sprays that you did. And these sprays are represented by these three arrows at the bottom. The y-axis, the canonical coefficient, can be thought of as the abundance of this community. And normally, curves with low amplitude are not different from the control. So here in this example, there was no difference between this treatment, this curve in yellow, and the control. Therefore, there was no impact of this treatment on the community abundance. Now, next, let's take a look at some real data. So have 2017 at the top and 2018 at the bottom. And our control is going to be the large plot size. So here is median against large, and there was no effect of plot size in both years. And here is a small against large, and again, no impacts on the of plot size on the arthropod community. Therefore, our conclusion was that plot size did not impact the arthropod community, composition, and abundance. Well, many of you know that some insecticides don't kill insects outright. They cause sublethal effects. And these sublethal effects cannot be measured by individual abundance or community abundance. You need to measure infield mortality directly to quantify those. So that's what you did. So you circle hundreds of Wi-Fi names and eggs, and you measured all sources of mortality in the field. That includes predation, dislodgement, parasitism, and etc. So let's take a look at some data. So here I have total mortality for eggs. Total mortality runs from 0 to 1, so 0% of mortality, 50% of egg mortality, and 100% of mortality. Here's a violin chart, basically shows the full distribution of the data, and the white circle represents the mean. So here's the data for large. Here it's medium, and here is small. And there was no effect of plot size on mortality of eggs. I'm showing you here only the data for eggs, but even for nymphs, there was no effect of plot size. So in conclusion, individual abundance, no effect of plot size. Community, no effect. And biological control, no effect of plot size. Therefore, our conclusion was that in small plots, 12 rows by 40 feet, are large enough for doing pest management research in Arizona. This research is particularly important for Arizona cotton because we rely a great deal on the natural enemy conservation as a key element of our IPM program. And of course, this supports target pest control, but how about the target pests themselves? Well, our research showed that 12 rows by 40 feet is also large enough for doing research with ligas and whiteflies. Here's an example of a 12-row trial, and you can see clear differences between the check and a carbine rotation. You are running the largest plots in the cotton belt. And here's another example, sulfoxaflor, a fully selective insecticide against a check. And you can see these clear differences in treatment because we are doing research at the right scale. So you can measure this insect response to treatment. And here's another example. This was a Ligas threshold trial. And again, you can see these differences in treatment. But how about whiteflies? Here's a video of Peter walking a 12-row Wi-Fi plot. He's holding a picture frame that got sprayed with canola oil to see how many whiteflies he could get on the frame by walking different treatments. And these are the results. So to the left, you have an untreated check that got disrupted with acephate, and you can see lots of whiteflies. And to the right, you had another treatment that followed the predator threshold. There are no sprays, and you can see clear differences between these two treatments. So 12 rows by 40 feet is also a large enough plot for whitefly research. So I go back to this figure that illustrates key elements in our IPM program, the selective technologies, and our key whitefly predators. And all of our recommendations depend on doing research at the right scale. If plot size is inappropriate, our recommendations might not hold up on commercial scales. This is why we invest so much time and resources investigating this basic question. This is to ensure that our research trials are emulating real-world conditions and that our recommendations are reliable. Thank you very much for your attention, and I will be glad to answer any questions that you might have.